Welcome to another episode from the Veterinary Medicine Digest. Today, we're diving deep into the fascinating, complex world of animal emotions, specifically focusing on our porcine pals, pigs. For too long, the inner lives of farm animals have been largely overlooked, but cutting-edge scientific research is now shedding light on their capacity to feel joy, fear, stress, and even empathy. Understanding these emotions is not just about curiosity. It's a crucial step towards ensuring better welfare and creating more harmonious relationships between humans and the animals in our care. So, get ready to have your perceptions challenged and your understanding expanded as we explore the emotional landscape of pigs. Our journey begins with a fundamental question: can pigs truly feel and share emotions, a concept we often attribute only to humans? Researchers have explored this through the lens of emotional contagion, a simple yet powerful form of empathy where one individual's emotional state influences another's. Imagine a situation where the cry of one infant causes other infants nearby to start crying too. They're sharing the distress, even if they don't fully grasp the reason behind it. This basic sharing of emotional states is considered essential for group bonding and communication, playing a vital role in survival by, for instance, alerting an entire group to present danger. It's also believed to be the phylogenetically oldest level of empathy, making its presence in many animal species, not just humans, highly probable. One pioneering study delved into this very phenomenon in pigs, using a clever experimental design to induce distinct emotional states and then observe their spread. Researchers worked with training pigs who were exposed to either a positive or a negative treatment. The positive experience sounds quite delightful: paired access to a spacious compartment brimming with peat, straw, and even a few chocolate raisins. Picture the joyful rooting and exploring. Conversely, the negative treatment involved social isolation in a small, empty compartment, combined with unpredictable, aversive handlings like a person restraining them with a nose sling or loud noises from a vacuum cleaner or bursting balloons. The behavioral differences observed in these training pigs were clear indicators of their emotional states. During the positive treatment, they displayed behaviors like playful running, gambling, pivoting, and shaking their heads with straw, along with excited barks and frequent tail wagging, behaviors universally associated with positive emotions. On the other hand, the negative treatment elicited responses like standing alert, attempting to escape, urinating, defecating, and holding their ears backward, often accompanied by high-pitched vocalizations and more low-pitched grunts, all recognized signs of a negative emotional state and distress. This confirmed that the experimental setup successfully induced the intended emotional responses. To test for emotional contagion, naive pigs, untrained pen mates, were then introduced to the test room alongside the training pigs, observing them as they underwent these treatments, but without experiencing the treatments themselves. What did the researchers find? Emotional contagion indeed occurred, particularly during the negative treatment. The naive pigs spent more time standing alert and tended to hold their ears backward when their training companions were experiencing the negative situation, compared to when they were in the positive treatment or alone. Standing alert and ears back are behaviors linked to negative emotional states, suggesting the naive pigs were indeed affected by their companions' distress. This spread of emotion could have been communicated through auditory or olfactory signals, as the pigs couldn't see each other. Interestingly, the study also hinted that the presence of naive pen mates might have offered a form of social support to the training pigs, making them less negatively affected during the difficult situations. The researchers also explored the role of oxytocin, a peptide traditionally associated with social bonding and emotional processing. It was hypothesized that oxytocin, administered intranasally to the naive pigs, would amplify emotional contagion. Surprisingly, the oxytocin did not directly affect the behavior of the treated naive pigs themselves. However, it did subtly influence the behaviors of the training pigs who had not received the oxytocin. For instance, training pigs were less likely to defecate during the negative treatment when their naive pen mates had received oxytocin compared to a placebo and showed different exploratory behaviors in the positive treatment. This suggests a fascinating, indirect role for oxytocin in pig communication, possibly by stimulating vocalizations or pheromone production in the treated pigs, which then affected their untreated companions. This interindividual effect of oxytocin is a concept that warrants much more investigation, as it could reveal deeper layers of social communication in pigs. Moving beyond the direct observation of emotional contagion, scientists are also exploring how environmental factors, such as music, can influence the emotional lives of pigs. We know that in humans, Music is a powerful tool for emotional communication, capable of inducing a wide array of affective experiences and influencing mood and behavior. But what about animals? Research into non-human models is vital for understanding emotions from biological and evolutionary perspectives. A compelling study investigated whether pigs could emotionally respond to musical stimulation and how those responses might relate to the music structure. Pigs, after all, possess auditory sensitivity comparable to primates and can distinguish between tones of different frequencies. This makes them an excellent model for exploring music's emotional impact. The researchers used a method called qualitative behavioral assessment, or QBA, which involves trained observers evaluating an animal's emotional states by integrating information from their body language, focusing on how the animal behaves, rather than just what the animal does. The study involved exposing nursery pigs to 16 distinct instrumental musical pieces, carefully composed to vary in their harmonic structure. Some were consonant, designed to evoke calmness. Others were dissonant, intended to induce fear or anger. And a third group had no harmony, being monodic with a single instrument. These musical sessions were compared to periods without music, including a habituation phase, breaks between pieces, and a final period. The results were quite remarkable. When pigs were exposed to music, both their positive emotions index, characterized by states like content, playful, sociable, and happy, and their negative emotions index, including fearful, inquisitive, and uneasy, significantly increased compared to periods without music. This demonstrated that pigs absolutely respond emotionally to musical stimulation, and these responses can be quite intense and diverse. Notably, fearful states were observed exclusively during the musical treatment periods, even absent during the breaks immediately following the music, directly attributing these negative responses to the musical stimulation itself. The most intriguing finding related to the specific type of music. Consonant music, with its stable and harmonious patterns, was strongly associated with positive emotional responses, grouping with higher values for the positive emotions index. Conversely, dissonant music, characterized by clashing and unstable patterns, elicited higher values for the negative emotions index, often leading to fearful and uneasy states. Music without harmony, using a single instrument, tended to be associated with more neutral states like calm, relaxed, and indifferent. This suggests that pigs, like humans, possess an innate or learned understanding of these harmonic structures and their associated emotional qualities. The study concluded that music has significant potential as an environmental enrichment tool for pigs. It promotes activity and exploratory behavior and can even generate mood swings that persist after the stimulation ends. This is important because environmental enrichment is crucial for animal welfare, allowing animals to perform motivated, species-specific behaviors. While previous studies on music and animal welfare had inconsistent results, this research provides strong evidence that carefully selected music, particularly consonant harmony, can contribute to positive emotional states in pigs and enhance their quality of life. This opens doors for designing musical stimuli specifically adapted to pigs' auditory characteristics and communication codes to promote desired emotional and behavioral responses. Beyond abstract sounds, the physical presence and interaction with a familiar human can also profoundly impact a pig's emotional state, a concept explored in another fascinating study. In intensive farming systems, providing enrichment is legally mandated in some regions, with manipulable materials being common. But can a human, through positive interactions, serve as a form of social or pseudo-social enrichment, fulfilling a similar role to an inanimate object? The answer, it turns out, is a resounding yes, and perhaps even more. This study compared pigs' perceptions of a familiar human experimenter with a simple, manipulable inanimate object, a water-filled plastic can with chewable pipe pieces. Both the human and the object were introduced to the pigs through a similar familiarization process involving repeated, positive contact sessions. For the human, this meant gentle tactile interactions, soft stroking, scratching, and speaking in a soft voice, actions known to promote positive states in pigs. First, a choice test was conducted, giving individual pigs the opportunity to express a preference between the human and the object by observing how much time they spent near or in contact with each. Surprisingly, the pigs didn't show a strong, consistent preference in terms of total time spent in proximity or contact. However, subtle but significant differences emerged. Pigs were more often observed to approach the human zone and, strikingly, they only chose to lie down when near the human, never near the object. Lying down is a powerful behavioral indicator of a lack of stress and a relaxed, positive state in pigs, suggesting that the human's presence had a reassuring effect. The real emotional depth was revealed in isolation/reunion tests. Here, pigs were first isolated for 5 minutes, a known stressor, and then reunited with either the human, the object, or remained in isolation, no stimulus. The researchers meticulously analyzed both behavioral responses and vocalizations, particularly grunts, which are rich in emotional information. Behaviorally, reuniting with a human significantly decreased the pig's time spent in an attentive, vigilant state and reduced their overall mobility to a greater extent than reuniting with the object or simply remaining isolated. This indicated that the human's presence was more effective at alleviating stress responses. While the object was attractive to some extent, pigs reunited with it behaved similarly to those who remained completely isolated in terms of postural and locomotory behaviors. The vocalization analysis further solidified these findings. Grunts produced by pigs reuniting with a human were significantly shorter in duration. Shorter vocalizations are consistently associated with positive emotional contexts in many species, including pigs. When comparing these grunt durations to those from other studies, the values observed with the human were well within the range of positively perceived situations. Additionally, reuniting with both the human and the object led to grunts composed of higher frequencies and a noisier spectral component, an effect that was even more pronounced with the human. These spectral changes are often linked to higher arousal in mammals. In essence, the study concluded that the familiar human, through their reassuring presence and previous positive interactions, could alleviate the distress of isolation and induce a high arousal, positive emotional state. The inanimate object, while interesting, did not offer the same level of emotional comfort and led to a more low arousal, negative emotional state, much like continued isolation. This highlights that promoting social or pseudosocial enrichment with humans can be a highly valuable strategy for enhancing pig welfare, potentially even more so than just inanimate objects, as humans offer dynamic, unpredictable, and interactive stimulation that objects cannot. To further objectively quantify these positive emotional states, researchers are turning to physiological measures, moving beyond just observing behavior. One promising avenue is the study of play behavior and its impact on an animal's heart rate variability, or HRV. Play is widely recognized as inherently rewarding for animals, inducing pleasurable states and promoting social skills, cognitive development, and even disease resilience. The question is, can we measure this internal positive feeling physiologically? HRV serves as a proxy measure of the autonomic nervous system's activity, reflecting the delicate balance between the sympathetic, fight or flight, and parasympathetic, rest and digest, branches. Emotional perceptions can cause shifts in this balance, with higher vagal tone, a measure of parasympathetic activity, often assessed by the RMSSD parameter of HRV, being associated with more efficient autonomic control and, crucially, positive emotions. While much research on HRV has focused on negative stimuli like fear or stress, which tend to decrease vagal tone, evidence is growing that positive emotions can actually increase it. A study investigated how play influences heart rate and HRV parameters in pigs. Gilts, or young female pigs, were chosen from groups that had received regular play opportunities from a young age. For the experiment, play was actively encouraged during 15-minute sessions, either in their home pen or in a larger playpen area with novel, destructible objects like cardboard, straw, or ropes. HRV was then measured during 3 consecutive periods, a baseline before play, during the play bout itself, and after play, immediately following a play bout. The findings were a significant step forward in objectively assessing positive emotions in pigs. The RMSSD, indicative of vagal activation, was significantly higher during play and remained elevated during the afterplay period compared to the baseline. This strongly suggests that engaging in play contributes to positive emotional states in pigs, and these rewarding feelings persist even after the active play ceases. Another HRV parameter, the SDNN, which reflects a more generalized sympathovagal balance, also increased during and after play, but with an important nuance. This increase was only observed when the pigs were actively moving forward, what the researchers termed ambulation. Heart rate itself increased during ambulation, but not specifically during play compared to baseline. This highlights a crucial point. Physical activity can confound HRV measurements. The energetic and excitable movements characteristic of play can activate the sympathetic system, reducing HRV. However, the sustained elevation of RMSSD even during movement suggests a co-activation of both the sympathetic, indicating arousal from the activity, and parasympathetic, indicating the positive valence or pleasure systems. This implies that more dynamic and energetic play, involving active movement, might actually be more rewarding for pigs due to the increased arousal it provides. Interestingly, the specific environment where play occurred, whether in the home pen or the larger playpen, did not influence the HRV measures. This is a valuable practical insight, suggesting that the positive effects of play can be achieved in various settings, which is good news for incorporating play opportunities into standard farming practices. This research provides compelling physiological evidence that play is indeed a source of positive emotion for pigs, further underscoring the importance of incorporating such rewarding experiences into their lives. Finally, as we strive for a more complete picture of pig emotions, researchers are exploring molecular markers that could provide objective and easily measurable indicators of emotional states. This led to a study investigating microRNA, or miRNA, as potential biomarkers of emotional valence in pigs. miRNAs are tiny, non-coding RNA molecules that play a crucial role in regulating gene expression throughout the body, including in the brain, and they've been implicated in emotional processing in humans and rodents. The exciting prospect is that these molecules can be measured in accessible bodily fluids like blood, making them ideal candidates for objective, non-invasive welfare assessments in a production environment. The study aimed to identify if specific miRNA patterns in the brain and blood would change during positive emotional states, validating these against established behavioral and physiological indicators. To do this, pigs were allocated to 2 different housing conditions for 5 weeks. Enriched housing or barren housing. The enriched group lived in groups of 3, received daily positive human interaction, and had access to a variety of toys like tennis balls, chains, and rubber matting, chosen to stimulate their senses and promote species-specific behaviors. The barren group, designed to induce a less positive state, was individually housed in barren stalls, with no human contact beyond feeding and cleaning, and no additional enrichment. As a behavioral measure of emotional state, the researchers employed a judgment bias test, or JBT. In this test, pigs were first trained to associate a blue bowl with a positive reward, chocolate treats, and a red bowl with an aversive experience, no reward plus a human scare. Once they learned this discrimination, they were presented with an ambiguous black bowl placed in between the familiar cues. The assumption is that if a pig is in a positive emotional state, it will be more optimistic and approach the ambiguous cue as if expecting a reward, whereas a negative state would lead to a more pessimistic response, approaching it more slowly or not at all. The results of this study presented a mixed bag, offering both unexpected findings and important insights. Surprisingly, the difference in housing conditions, enriched versus barren, had no discernible effect on the pig's performance in the judgment bias test. This meant that, behaviorally, the pigs in the enriched housing didn't show a significantly more optimistic bias than those in the barren housing. Furthermore, and perhaps even more unexpectedly, there were no significant changes in miRNA expression patterns in either the blood or the brain, specifically the amygdala, between the 2 treatment groups. However, the study did reveal a crucial physiological difference. Pigs in the enriched housing showed a significantly increased concentration of dopamine and its metabolite, dopac, in a brain region called the striatum, compared to pigs in barren conditions. Dopamine is a key neurotransmitter strongly implicated in reward processing and motivation, so this finding is consistent with the hypothesis that enriched housing indeed promoted a more positive emotional state at a neurochemical level. Yet, this increase in dopamine was not observed in other brain regions like the amygdala or prefrontal cortex, nor were there any differences in serotonin levels. The lack of change in miRNA profiles and the behavioral judgment bias was discussed extensively by the researchers. They proposed several possible explanations. First, perhaps the environmental manipulations, enriched versus barren housing, were not extreme, enough to induce a sufficiently strong or sustained change in affective state that would be reflected in miRNA expression. miRNA changes might be more indicative of invasive physiological stressors like heat or pain rather than more subtle environmental differences. Second, there might have been issues with the design of the judgment bias test itself, such as pigs learning that the ambiguous cue was never rewarded, which could have confounded the results and led to false measures of optimism or pessimism. They also suggested that the type of enrichment chosen might not have been optimally rewarding for the pigs, or that social competition in the enriched groups could have introduced negative experiences. Despite the null findings for miRNA as biomarkers under these specific conditions, the study underscores the ongoing challenge of developing objective measures for positive emotions in animals. The observed increase in dopamine provides a compelling reason to continue exploring the biological underpinnings of positive welfare, even as we refine our behavioral and molecular assessment tools. It highlights that the absence of evidence for a biomarker doesn't necessarily mean the emotional state isn't present, but rather that our methods for detecting it may need further refinement and validation. So, As we bring together all these incredible threads of research, what have we learned about the emotional lives of pigs? It's clear that these intelligent animals possess a rich and nuanced emotional landscape. They can experience and even share both positive and negative emotions, a phenomenon observed through emotional contagion. Their behaviors, from playful tail wagging and barks to alert standing and distressed vocalizations, reliably indicate their internal states. Furthermore, external stimuli profoundly influence these emotions. Consonant music can bring them joy and relaxation, while dissonant sounds can induce fear. Perhaps most compelling is the power of the human touch. Positive interactions with a familiar human can provide profound reassurance and induce genuinely positive emotional states, going beyond what even attractive inanimate objects can offer. This pseudosocial enrichment with humans can alleviate stress, induce high arousal positive states, and even alter vocal characteristics in ways that clearly signal pleasure. And when pigs engage in play, we now have physiological evidence from their heart rate variability that they are indeed experiencing positive emotions, with dynamic play potentially being even more rewarding. While the quest for perfectly objective molecular biomarkers like miRNA is still ongoing, with research showing challenges and the need for more refined approaches, the discovery of increased dopamine in enriched pigs reminds us that even subtle environmental changes can have a real impact on their neurochemistry. This collective body of research paints a vivid picture of pigs as sentient beings with complex emotional lives. It reinforces the critical importance of animal welfare, urging us to consider not just the absence of suffering, but the presence of positive emotional experiences. From providing engaging environments, carefully selected music, positive human interactions, and ample opportunities for play, we can profoundly enrich the lives of pigs. The future of veterinary medicine and animal husbandry lies in this holistic understanding, combining behavioral observation, physiological measurement, and the search for molecular indicators to create optimal living conditions for these remarkable animals. This deep dive into pig emotions truly showcases how science is transforming our understanding and relationship with the animal kingdom. Thank you for listening. The titles for the reference papers are in the video description. From Vet Med Digest, where we keep minds inspired, hearts light, and tails wagging.